So we are halfway done. We are about to start Unit 3. Unit 3 and Units 3 and 4 are my favorite of the semester. Unit 3 is all about genetics. So we start with cell division, we move into classical genetics or Mendelian genetics, which I find students really like and are usually familiar with from high school. And then chapter 12, we talk about chromosomes, most specifically sex chromosomes, the X and Y chromosomes, and the particular type of inheritance patterns that we see in those chromosomes. We continue genetics in Unit 4 with DNA replication, so copying and passing on the genetic material in an organism. And then chapter 4 is gene expression, so how we go from a gene all the way out to the physical expression that we see. And then the rest of Unit 4 is our evolution section, which is always quite popular as well. We start out talking about Charles Darwin and the idea of natural selection. Then we move into how genes affect evolution and how evolution acts on the genome of an organism. And then if we have time, sometimes we do, sometimes we don't, we will get into speciation in chapter 22, which is how you move from a population changing to a population changing so much that it becomes its own species. So let's get started with chapters 9 and 10. Chapters 9 and 10 focus on the cell cycle. And then we talk about two different types of cell division, mitosis and meiosis, and how that relates to the sexual life cycle of most organisms, animals and humans. So to get started, let's review the cell theory. So we talked about the cell theory back in chapter 1 a little bit, chapter 4 a little bit, and the cell theory states that all living cells are made up of one or more cells. So that's a fancy way of saying that everything that's alive is made up of at least one cell. So our prokaryotic organisms are made up of one cell. Those are our bacteria and our archaea, and some of our eukaryotes, our protist kingdom. And then other animals are made up of more than one cell. So these can be simple multicellular organisms or really complicated multicellular organisms such as mammals. But the point is that the cell is the smallest unit that we call living. And we talked about why that was. We talked about some of the characteristics of the cell. One is that it has to pass on genetic information. It also interacts with the environment, exchanges energy and chemicals. It interacts with other organisms. And there are a variety of other things that we consider cells doing or experiencing that suggests that they are in fact the smallest unit that can function as its own living organism. But there's a third part of this cell theory that we haven't really discussed yet, and that is that all living cells come from existing cells. So this theory was put forth in the mid-1850s by a man named Rudolf Virchow. And Virchow was a German pathologist, so he was studying disease and he was studying organs and tissues, and he was trying to figure out where infections came from. But he proposed that cells don't just sort of spontaneously appear. They must come from a pre-existing cell. And they have to do that by dividing somehow. So the ability to pass on genetic information is very tightly linked with cell division. So if an organism is going to live and if an organism is going to reproduce and pass its genetic information on to the next generation, it has to copy its genetic material and it has to pass it on in a very specific way so that all of that material makes it to the next generation. And this is important if you are a growing and developing cell that's going to turn, group of cells that's going to turn into an entire organism, all of the cells in that organism have to get the correct genetic information. And then it's also important, if you are reproducing, you have to pass on your genetic information to the next generation. And so that's what we're going to talk about in this chapter. So we start with chapter 9, and we do chapter 9 and 10 as kind of a quick unit. Because they're very closely related, a lot of the concepts overlap, and I do kind of talk about them interchangeably, and your assignment will be very much overlapping and comparing and contrasting the different types of cell division. But we first talk about the cell cycle and mitosis, which is cell division of everything but the cells that are going to make sperm and ovaries. So cell division is really important for passing on genetic information to the next generation. If you don't pass on all of the genetic information and you don't pass it on correctly, that next generation of cells is not going to survive. If you are a prokaryote, you generally divide by something called binary fission, and we're not going to talk about what that is. It's really similar to the process of mitosis, which we are going to focus on. But the important thing here is that if you're a prokaryote and you divide, you are producing an entire another organism. So we have a really simple picture here. This is a single cell with a single piece of DNA. That DNA is copied. We will get to that in chapter 13 and learn how the cell actually copies all of its DNA in an orderly fashion. And then that DNA is partitioned into what will become the two new cells, and then eventually you get those two daughter cells. So the parent cell splits in half and produces two identical daughter cells. So if you're a eukaryotic cell, we'll just say the process of mitosis is really important in producing an entire organism. But we also have eukaryotes. We are eukaryotes. Most of the things that we think of when we think of living organisms are actually eukaryotes. And eukaryotes perform cell division too. Eukaryotes perform two different types of cell division. One is mitosis, or reproduction of the somatic cells. So somatic cells are, if we're talking about humans, all the cells in our body that are not the cells that produce sperm or eggs. So there are skin cells, our brain cells, our neurons, our all the cells in our organs, all of those different types of cells produced by a process of mitosis. And so most of the cell division that's going on in our body is actually mitosis. And then we have a specialized type of cell division called meiosis. And meiosis is used to produce the gametes, so the sperm and the egg cells. And it's a slightly different process. The general idea is the same, but there are some key differences that make it specific for producing those sex cells. So if you are a eukaryotic cell, there are several reasons that you would divide. One is reproduction. So there's reproduction of the cells that are involved in actual fertilization, the sperm and the egg cells. So this is reproduction of an entire organism. There's also reproduction of the cell just to produce new cells. And that is really important in growth and development of the organism. So if you think about an egg that has been fertilized by a sperm, now it has to divide, it has to grow into a full-blown organism. And all of that takes place by cell division. So cell division allows for growth and it allows for different development. Development happens when those cells that have divided differentiate into their different jobs within the organism. And that happens really early on in development, but it happens at specific times. So a cell will divide and divide and divide, and at a certain point, some of those cells will specialize. And that leads to the development of the different tissues and the different organs and things in a living organism. And then finally, we have tissue renewal. So this is basically once your growth and development has finished, or even while it's going, some of those cells die and they have to be replaced. So think of this as if you get a cut on your arm, or if you have dry skin and some of your skin sloughs off, all of those cells have to be replaced. Or if you have damage to some sort of organ or tissue in your body, those cells have to divide to repair and replace those that are damaged. And then, of course, sometimes cells just wear out. Cells have a limited lifetime. They perform their job for a certain amount of time, and they just need to be replaced. So while cell division in prokaryotes, basically the sole purpose is to produce a new organism, there are a variety of purposes that cell division serves in a eukaryotic organism. So the first type of cell division that we're going to talk about is somatic cell division. So that is the process of mitosis, producing all of those cells in the body that are not the sperm or the egg. So most of the cell division occurs in those somatic cells. And one of the key features of this cell division in mitosis is that all of the cells that are produced from mitosis are genetically identical, or they should be genetically identical. And if you think about it, that makes sense, because if you are a skin cell, you need to be a skin cell when the cell differentiates, or I'm sorry, when the cell divides. And so one skin cell has to pass on all of the correct information to that next skin cell so that it can work and do its job. If you're going to divide, the first thing that you need to do is replicate the DNA. You have to copy the DNA and distribute a copy to each cell. So that DNA has to be replicated faithfully. All of the genetic information of an organism is contained in every single cell. So if you're talking about humans, that's 46 chromosomes, 46 very large pieces of DNA that have to be copied every time the cell divides. And then it has to be divided. And you have to divide it carefully. You have to make sure that each cell gets the correct number and correct complement of chromosomes. Because if they don't, the cell's not going to survive. The cell needs all of its genetic information. If that DNA is not faithfully replicated, you can have problems. You can have things pop up like cancer, or just cells that aren't functioning correctly in an organism. And then you're not going to have a healthy organism. So we're going to start our discussion of mitosis by talking about the cell cycle. So the cell cycle is just the life of the cell from one division to the next division. When we talk about this, we use family terms. So the parent cell is going to divide into daughter cells, and those daughter cells are going to be genetically identical to each other and should be genetically identical to the parent as well. So all of our genetic information is organized in a very specific way in our genome. A genome is just a fancy word for all of the genetic information in our cells. So if you're a prokaryote, basically you have one circular piece of DNA, or at least one large circular piece of DNA. They often have little tiny pieces of DNA as well called plasmids. Those are not necessarily replicated in the same way that a singular circular chromosome is. So if you're only replicating one piece of DNA, replication is not super complicated. Cell division is not quite as complicated. But if you're a eukaryote, you have multiple long pieces of DNA. Humans have 46 chromosomes, chimpanzees have 48, chickens have 78, bananas have some crazy number, strawberries have some crazy number of chromosomes. And so you have to keep that genetic information organized in a very specific way. One, so that genetic information can be expressed in the cell, and two, so that it can be divided properly during cell division. So all of that DNA is organized into chromosomes. So a chromosome is just a long piece of our DNA molecule. We talked about really quickly the structure of DNA back in chapter 3, and we'll come back to more specifics about the structure of DNA and the structure of chromosomes a little bit later. But for now, it's enough to say that in eukaryotes, we have multiple pieces of long, straight chromosomes, or long, straight pieces of DNA. On each chromosome, there is genetic information from anywhere for 100 to 1,000 or a couple of thousand genes. And then mixed in with that is some non-coding regions, some regions that are special in their structure. But not all of the genetic information is actually coding for something. But contained within that sequence of DNA in that chromosome, you do have several hundred genes on each chromosome. So we have a couple of different pictures here. On the top right, we have a cell, and those purple squigglies over there are the chromosomes. Those are slightly condensed chromosomes. Down below that, we have what we call sister chromatids, and we'll come back to that in just a minute. But that structure there, that X-shaped structure, is probably what you're most familiar with when you see a chromosome. And that is actually two identical copies of DNA bound together at the center, or the centromere of that chromosome. And it's all kind of tightly packed and squat and stout because it is condensed into something called chromatin. So chromatin is a complex of the nucleic acid, the long chain of nucleic acids, and proteins. And the reason for this is that those chromosomes are wrapped around specific types of proteins called histones. And those histones allow that genetic material to be sort of wound nice and tight, to keep it organized, and also to tightly pack it. There's something like 6 meters of DNA in every single cell, and it's packed in there because of all of this really tight packing and folding. You can visualize this if you have a shoelace between your fingers, and you twist one end and you keep the other one straight, and you keep twisting and twisting and twisting, and eventually it forms these supercoil loops that are loops folding back on loops. And if you keep twisting, it'll condense almost into a little knot. And that's what's going on, except that in that analogy we don't have proteins. But those proteins are to help organize the DNA into this very specific structure. So let's talk about those chromosomes and chromatids. There are quite a few terms in this chapter that are going to sound really familiar, or I'm sorry, not familiar, sound very similar. So chromosomes are a single piece of DNA. On the last slide, we talked about chromatin with an N at the end. That is our tightly condensed DNA molecule wrapped around proteins. Then we have chromatids with a D at the end, which I also mentioned. Chromatids are duplicated chromosomes that are condensed and attached. So it is a single chromosome that has been copied but kept together at the centromere. So the centromere is sort of that waistline we see in this chromosome. So down here, I'm down here in the middle of this picture. We see this almost X-shaped piece of DNA. One side of this, the left-hand side, is one full chromosome. The other side is its exact copy. So it's basically like you have taken a photocopy of that chromosome. This little pinch in the middle, that kind of looks like a waistline, that is the centromere. So that's a structural part of the chromosome. It's super condensed there, and its purpose is to hold these chromosomes together, and also to, we'll see this again later, to allow for attachment of some special structures that are going to attach there and then pull these chromatids apart. So when we get to mitosis and meiosis, we have to keep track of the number of chromosomes, and I'll talk about why that's important later. But we count the number of chromosomes based on the number of centromeres. So up here at the top, we have our single chromosome. We've got our centromere in the middle. This short arm up here is called the P arm for petite. So it's the small arm. And then the other arm down here is called the Q arm, for no other reason than Q comes after P, and they picked P for petite. So anyway, we see that centromere in the middle. So this molecule, I actually point to this one over here now. This DNA molecule is going to undergo DNA replication. And so within that cell, it's going to copy all of its chromosomes. So every single chromosome is going to be copied, and it's going to be kept together with its identical sister, its chromatid. So sister chromatids are basically twin sisters. They are identical copies of each other attached at the hip. So over here on the right hand side, we see the duplicated double helix there. And then during the process of cell division, those chromosomes are going to be pulled apart and separated into daughter cells. So at the end of mitosis, all of the cells should have exact copies of all of its chromosomes. We only have one chromosome to start with. You can see that very easily, one chromosome here. But we count our chromosomes based on the number of centromeres. So when we get down here to number 2, we have sister chromatids. But think about what I just said. We count chromosomes based on number of centromeres. So how many chromosomes do we have there? When we have sister chromatids, they are considered one chromosome. They are two pieces of DNA, but they are attached and considered one chromosome. So between steps 1 and 2, we only have one chromosome, even though that DNA has been replicated. When we finish mitosis and we split those sister chromatids up, we give each sister chromatid to a new daughter cell, we now have 2 chromosomes. So when they pull apart, we now have 1, 2 centromeres, and so 2 chromosomes. So let's recap a couple of terms. The first was somatic cells. So those are all of the cells in the body minus the sperm and the eggs, and they undergo the process of mitosis, where each cell is genetically identical to the parent and to its sister cells. Excuse me. In this picture up here, we have our parent cells. Down here we have the 2 daughter cells. They are also called sister cells because they are the next generation of cells together. And then a term that I didn't actually talk about, diploid. I'll come back to this, especially when we talk about meiosis and sexual reproduction. Diploid means that a cell has 2 copies of each type of chromosome. So those chromosomes come from one from mom and one from dad, one in the egg and one in the sperm. So if you think about an egg cell, it's going to have 23 chromosomes from mom, a sperm cell 23 from dad, come together. Now there's 2 copies of all of those chromosomes. And so humans are diploid with 46 chromosomes, 2 copies of 23 different types of chromosomes. We'll come back to that, but I just wanted to introduce that term because we're going to use that when we talk about meiosis.